Welcome, and thank you for joining this webinar on PFAS and contaminated sediment sites. My name is Miranda Henning. I am a Managing Principal and Business Director at Integral, and my work largely focuses on toxicology, health, and ecological sciences, very often related to contaminants in sediments. I'm pleased to be joined today by my colleague, Jared Gasper, who has more than 15 years of experience related to the fate and transport of PFAS and other organic contaminants, as well as metals, at both land-based and aquatic sites. Rob Fox is also joining us. He's a named partner at the law firm of Manco Goldcatcher Fox. Go ahead. We'll spend about 15 minutes each on 3 topics. I'm going to begin with the basics of PFAS chemistry, uses, properties, and why they're not your typical sediment contaminant. Jared is then going to discuss investigation strategies for PFAS at contaminated sediment sites. And Rob will discuss the regulatory and legal setting. Throughout, we will be drawing comparisons between PFAS and a legacy sediment contaminant, PCBs. We should have about 10 or 15 minutes at the end of our presentation to answer your questions. Feel free to type them into the Q&A at the bottom of the page as we go along. Okay, let's go ahead to PFAS 101. When we start thinking about PFAS, we want to understand what they are. So basically, they are synthetic organofluorine chemicals with diverse structures and applications. The fact that they're synthetic is important. They don't occur naturally. Often, PFAS are simply defined based on structure, as described here in the second bullet. Among the complications in defining PFAS is that the fact that PFAS are not limited to just the manufactured substances and their salts, but they also include the degradant precursors, metabolites, byproducts, and other transformation products. So this is in clear contrast with PCBs, the legacy sediment contaminants that I've spent much of my career thinking about. I distinctly remember working on my first PCB contaminated sediment site back in the early 1990s and thinking just how complicated PCBs were. How times have changed. With PCBs, we're only talking about 209 individual congeners. And all 209 congeners share a very similar structure. They have 2 phenyl rings, or a phenyl ring is a ring of 6 carbons and 5 hydrogens, and between 1 and 10 attached fluorines. Now, in sharp contrast with PCBs, there's no consensus about the definition of PFAS. So this next table shows 4 different organizations' definitions for PFAS, ranging considerably in breadth, such that we may only be referring to a few thousand distinct chemicals, such as those near the bottom of the page with the narrower definition, or we may be talking about more than 10,000, such as those at the top of the page. This table, by the way, is excerpted from the White House PFAS report, which was written for Congress and therefore is one of the more digestible treatments of PFAS for non-scientists. This and other really good sources of information on PFAS are available on URLs that you'll find on the resources tab of the Zoom meeting. So one way to understand the PFAS family is through this family hierarchy, which was drawn from ITRC's fact sheets, which are another really good source of information on PFAS that you can find links to on the resources page. So starting at the top, the PFAS family can first be divided between polymers and everything else. For the non-chemists in the audience, polymers are substances with molecular structures that consist mostly or entirely of large numbers of similar units that are all bonded together. Polymers are of particular interest here because their structure and their size make them less biologically active than the non-polymer PFAS on the left-hand side of this family. So the non-polymers are divided into two subclasses: perfluoroalkyl substances and polyfluoroalkyl substances. Jared is going to get to this in more detail shortly, but in the meantime, just as an intro, perfluoroalkyl substances are organofluorine compounds, that is, carbon fluorine compounds that lack carbon-hydrogen bonds. In polyfluoroalkyl substances, some of the fluorine atoms are replaced by other atoms, like oxygen and hydrogen. So again, to compare this family tree to that of PCBs, the PCB family tree would really just look like a single parent with 10 offspring that differ only in terms of the number of attached fluorines. So a really simple analogy is this is like a dog that has a litter of 10 pups, where each puppy has a different number of fluorines. Whereas PFAS are more like entirely different breeds of dogs. They come in all sorts of shapes and sizes. Some are short, some are long, some are straight, others are branched or curly-haired, like the Commodore here. And each were bred for different purposes. Along the way, we also get thousands of different kinds of mutts. All right, back to chemistry. So this is PFOA, or perfluorooctanoic acid. So the gray spheres on this illustration are carbon atoms that are linked in a chain, and because there are 8 of them, octane is in the name. The green spheres are the fluorine atoms that are bonded to the carbon atoms attached here to all possible bonding sites. So that means it's perfluorinated. The fluorine tail end is strong and stable, which gives it lipid and water repelling properties, but also makes it quite persistent in the environment. And finally, the red spheres are oxygen atoms, and the white sphere is a hydrogen atom that dissolves in water to make this an acid. So the many different PFAS substances reflect different histories of invention, the manufacturing process, and the uses. So near the top of this page, you see PTFE, which was invented first back in the 1930s and initially applied as nonstick coating for several decades before they were applied to fabrics for waterproofing starting in the 1970s. PFOS and PFOA came next in the 1940s. These were used to make stain and water-resistant products, firefighting foam, and protective coating through the 1990s. It's also worth noting that electrochemical fluorination, or ECF, was the dominant manufacturing process until the 2000s, when fluorotelomerization was introduced for the production of fluorotelomers, which became the predominant form of firefighting foam beginning in the 2000s. Those diverse uses are only possible because of their diverse properties. So we get wide-ranging structures and characteristics ranging from solids, liquids, and gases, inert to highly reactive, and mobile to mobile, and bioaccumulative and not. These diverse properties, These diverse properties mean they behave in a highly variable way in the environment, which in turn leads to considerable variety in their environmental data. So again, comparing them to PCBs, yes, the different PCB congeners lead to some differences in the degree of toxicity and persistence, but there's consistency across PCBs with respect to their general tendency to be hydrophobic, bioaccumulative, and persistent. You know, one difference between PFAS and PCBs that surprises some people because of the often used term forever chemicals for PFAS is that PFAS are actually more susceptible to degradation in the environment than are PCBs. So all this is to say, you can go ahead, that PFAS are not a typical sediment contaminant. The pathways by which PFAS enter aquatic systems is often different, than what you see for legacy sediment contaminants. So for instance, PFAS generally enter the environment via a release to soil, followed by leaching from soil to groundwater and discharge of the groundwater to the surface water. In addition, in contrast with legacy contaminants like PCBs that really like to hold on tight to the organic carbon that's in sediment, there is a tendency for PFAS to move to and stay to the air-water interface, whereas PCBs typically are bioaccumulated through the food chain as a result of consumption, the diet of the fish eating the invertebrates and then mammals and birds eating the fish. Here with PFAS, you see bioaccumulation in fish via gill transfer of surface water. And then finally, this is a really important point. We've got quite limited data on concentrations in sediment, water, and biota that are safe for both aquatic organisms and the people and the critters that eat those aquatic organisms. This too differs from the case of PCBs, where we generally have a reasonable understanding of safe concentrations in surface water, sediment, and tissue. In addition, with PCBs, we tend to see greater sensitivity to PCBs from ecological receptors as compared to humans. Whereas with PFAS, we tend to see, as best as the existing data can show us, a greater sensitivity of humans rather than ecological receptors. And with that, I will turn the stage over to Jared, who will lead us through investigation strategy.
Thanks, Miranda. That was a great introduction, and I'm going to discuss investigation strategies at sediment sites. I'm going to cover 4 topics, really. First is site evaluation, then we'll discuss some example CSMs and highlight some key factors pertaining to PFAS. Then we'll discuss a little bit about source identification. And finally, we'll dig into the meat and understanding the interactions between PFAS and sediments and some of the key factors and traits of PFAS that make PFAS and sediments an interesting situation. So to begin, this will look familiar, I think, to a lot of folks out there, is we've got to evaluate the site, be it an upland facility, be it a sediment site, be it a river, but we got to begin with a site evaluation, and in this evaluation, we use 4 pieces or groups of information that ultimately lead in to build a conceptual site model. Where the conceptual site model it is the one-page distillation of our understanding. And we'll talk about that more. But first, I want to talk a little about the information that we need to gather. The first is an understanding of site history. This is really the historic and current operations or facilities or processes that occur at a site. And that leads to our understanding of the use and release of PFAS at that site or that facility, where our goal here is to define the magnitude and the duration and the timing of those uses and releases, recognizing that for both we can have unintended uses and releases or intended uses and releases. For example, our intended uses might be the actual manufacture of PFAS or the use of PFAS in a production of a product or a process. We may also have unintended releases such as spills, or intended releases such as outfalls or air emissions. Then we need to take that information and start to intersect that with our understanding of the fate and transport pathways, how the PFAS is going to move through the environment, the air, the water, the sediments. And then we combine that information with the amount of data that's available for the environment. And one of the big differences here for PFAS is that we have a lot less data available. We have less data available spatially, we have less data available temporally, really because people have only been focused on these compounds in the recent past. And we use that information to build our conceptual site model, where the purpose of that is to build our understanding of what's going on and to identify the data gaps and the puzzles and the places where we need to develop a better understanding. So a couple example CSMs that highlight some interesting factors pertaining to PFAS. The first one is this watershed scale CSM, a really broad scale. And the first thing that we notice is that PFAS can be used and are present throughout land uses. So sort of starting at the top, we have rural and agriculture uses where PFAS may be used in such things as surfactants and pesticides. It may be used as an agricultural, or it may be present as an agricultural soil amendment of biosolids. It can be used or manufactured in industrial facilities. We can see the uses and the releases of PFAS at commercial operations. For example, here we have an AFFF site. We also have uses and releases of PFAS at residential facilities. This is sort of the result of the ubiquity of these products and these compounds in consumer products. And then at the bottom, we see both a municipal and a private water source. And heretofore, a lot of the focus of PFAS has been about the impacts on drinking water supplies. But we also recognize that sediments are a key link or a key piece of this CSM, linking multiple sources, linking the water and linking the other things. So our understanding of sediments and PFAS are key to understanding the fate and transport across this sort of watershed scale CSM. Then we can focus in and zoom in to a smaller, more focused CSM. For example, here we have a wastewater treatment plant, a publicly owned treatment works. And the first thing we need to recognize is that this type of facility is going to handle PFAS from a diverse range of sources, be it stormwater or waste effluent or domestic sewage. So these facilities end up really receiving these very diverse, complicated, and unknown mixtures of PFAS. And when we think about hydrophobic legacy contaminants such as PCBs that may end up in a facility such as this, the first thing that we know is that these compounds are very hydrophobic. They're going to sorb to the biosolids and leave the facility there. Very little to none is going to be discharged with the water. But one of the interesting things that we've seen in our project work, has been published in the literature before, is that at these wastewater treatment plants, oftentimes we see PFAS concentrations that are higher in the effluent than they are relative to the influent. And this is recognizing the fact that a whole lot of PFAS leaves as biosolids because of the sorption properties. In this sort of this puzzle, why is more leaving than is coming in, doesn't really make a whole lot of sense when you just look at it to begin. But what we're seeing is that there are transformation processes occurring across the wastewater treatment facility, and precursors are degrading and becoming known PFAS molecules. And so we see these increases in concentration. So these CSMs, what are they helping us with? What are they getting at? First thing is that these sources of PFAS to the environment can be diverse. They can be hard to identify. We really need to understand and measure the PFAS at these different points and understanding the magnitude of the different fluxes, and that we need to keep in our mind the fact that these compounds may transform and degrade in the environment and shift identity. And those are some of the key points to getting at solving the puzzles and the difficulties of understanding the fate and transport of PFAS. So to kind of discuss some source identification challenges that we see at facilities and eventually at sediment sites, is that a lot of these compounds are not listed on packaging or safety data sheets that you would receive when you purchase a product. Oftentimes they're proprietary, so no one's going to tell you what's in them. Oftentimes PFAS containing products are spec-based, where really the product is purchased for the properties that it imparts to something being produced at the facility. Um, and there's not much concern as to what it actually contains. We also know that a lot of these compounds are technical mixtures. They're not pure compounds. So we have various amounts of byproducts or reagents and reactants, which constitute a small fraction of the overall mixture. But we also need to recognize that when we're talking about PFAS, we're in the parts per trillion environment. And so small components within the technical mixtures may be important when we start thinking about part per trillion space. We know that the formulations have changed over time. This has primarily been a shift from longer chain PFAS to shorter chain. And they haven't been defined as hazardous substances in the past. And so we don't have a body of reporting information about the use and handling of these materials, such as we might for other compounds with the toxic release inventory. So if we don't know what the sources are, we have some gaps in our CSM, and then we need to start understanding more about, we need to measure things. So if we start talking about measuring PFAS, we really need to begin with determining what is the question we're asking. And there's kind of two ways of getting at that. The first question is, are PFAS present? Are any of this group of compounds present? The second way we can think about that question is an individual PFAS compound, be it, let's say PFOA or PFHXS, just to have a couple examples, is that specific compound present? Maybe a regulator is asking you that question, or the concern is about a specific compound. So if we're asking that first broader question of, are PFAS, the larger group, present, we can use kind of screening or semi-quantitative methods. The main one here is what's called total organic fluorine. And so this measures carbon bonded to fluorine. It gives you a broad measure of what's out there. One of the things that we know is that naturally occurring organofluorine compounds are essentially non-existent. So this kind of method would give you an idea of if there are any PFAS present at your facility or in your sediments. If you're asking the second question of, is there an individual PFAS molecule present, then we really need to focus on the quantitative methods. The most important one here is EPA method 1633. It can be used for water, soils, sediments. It's in draft phase. It'll get you to, about 30 or so PFAS, individual PFAS molecules. You may be in a case where you know a little bit more about your chemistry. Maybe your neighbor is doing something funny, you know something about their chemistry, and those other methods won't measure what you're interested in. Then you can talk to the lab about developing custom or proprietary methods to measure specific PFAS compounds. But as we kind of move from the organic fluorine to the EPA methods to custom methods, we're really moving from simpler to more complex and less costly to more costly. And then the last group of methods that are of interest here are the precursor methods. For example, there's one called the TOP assay, or the total oxidizable precursor. And in this method, you take a amount of sediment, you measure the PFAS in it, and then you take another aliquot of that sediment and you add a strong oxidant to it, and then measure the PFAS present after that oxidation. The purpose of this oxidation step is to get an indication of what degradation products, what degradation processes may occur in the environment. And so it gives you an idea of what the amount of transformation possible or potential in the environment may occur, recognizing that the conditions of the laboratory method are different than what we see in the environment. So in terms of transformation, what are we talking about? If we think about this, there's sort of the group of known PFAS. So if you get EPA method 1633, you measure 30 of them and you get a certain amount. But then there's this additional mass of precursors that we really don't have any idea in any given sample how much is out there. But to kind of demonstrate the potential importance of these precursors, on the right-hand side here, I'm showing a plot of the results of a top assay for some sediments from the Netherlands. The orange bars that we see, or you may not see, they're awfully small. That's the amount of PFAS that was measured in this sediment sample prior to the top assay. Then the large blue bars represent the amount of PFAS measured in the sample following the top assay. So what we're seeing is that there are many, many more times more precursor mass with the potential to produce known PFAS molecules than there are present initially in the sample. And so this is an indication. It's pretty evocative. It may not be representative of all sediment samples, but I think it really demonstrates the potential importance of transformation. And the point here is that, materials or sediments may not have PFAS in them now, but 6 months, a year, 5 years, 50 miles downstream, they may undergo transformation reactions and produce PFAS, known PFAS molecules. So if we dig into the kind of chemistry of this, let's say, for example, we have a facility that is utilizing PFAS-containing compounds to provide stain and water resistance to a fabric. And if we were to zoom in with a magical microscope to the surface of the textile, what we would see is that there's this PFAS-containing polymer that's actually used to provide the water and the stain resistance. And this polymer is made of a few pieces. One is a hydrocarbon backbone that provides adhesion to the textile. This is often such compounds such as a urethane. Then we have a linkage molecule, and that linkage molecule provides the linkage from the adhesive backbone to the actual fluorinated moieties. So at the end of these polymers, we have these fluorinated moieties that actually provide the stain or water resistant properties that you're interested in. And over time and in the environment, what happens is these polymers start to break down. And what the endpoint of these breakdown reactions are known PFAS molecules, where the identity of that PFAS molecule depends on what the initial polymer is. So as we have a degradation, the PFAS that's contained in these polymers becomes visible, but we also see a change in properties over time where we see these molecules becoming smaller, they're becoming simpler, the molecular weight's decreasing, and so we see decreasing absorption and increasing transport in the environment as these precursors are degraded. And this is just an example of one specific kind of precursors, but the breakdown process and the change in properties as this breakdown occurs is going to be analogous for other classes of precursors. And then in terms of source identification, we talk about 3 different primary characteristics that were alluded to earlier by Miranda. The first is chain length. The second is functional groups, and then it's linear versus branch. So if we looked at pictures of this, to begin with chain length, up in the upper part of this plot, we see PFBA, which is a 4-carbon perfluorochemical. We can see that this chain is short. It contains just 4 carbons. And then what we can have is increasing chain length, where this sort of ladder of the fluorinated chain becomes longer and longer and longer. Chain length also can give us some information. Generally, longer chains are older than shorter chains. The next major difference we have is functional groups. So if we compare PFBA in the upper part of the slide to PFBS in the lower left-hand side of the slide, they're both 4-carbon fluorinated chains, but the functional group at the end is different, where the BA has a carboxylic functional group. The BS has a sulfonate functional group. This can provide you some information about the application of the material or the manufacturer. The last difference that we can use to identify sources is this linear versus branched. So in the lower right-hand corner of this plot, what we see is a branched PFOS molecule. So you can see at the fifth carbon, there's actually a branch as opposed to a straight chain. And the branched versus linear can give you an indication of which manufacturing process was used to produce the chemical. So these factors, not only do they tell us about the source, they allow us to differentiate the sources, but these structure differences are also key to understanding fate and transport in the environment. So for PFAS in the environment, the structure is important because it's really, the structure is the destiny for fate and transport. And as a result, not all compounds will accumulate or remain in the sediments. This was demonstrated really well by some work done by Chen et al. In 2016. They did some laboratory studies where they took some water, they took some sediment, they took some PFAS, they mixed it all together in a vial. And then after they allowed it to equilibrate, they split it into three groups, three kind of parts of distribution parts. The first part is the aqueous phase. So this is represented by the blue in the plots. This is PFAS that's just dissolved in the water. The second phase is sorbed PFAS. So this is PFAS that is sorbed to the sediment, but will leave relatively rapidly and relatively easy. On the plots to the right, this is demonstrated by the gray part. The third phase is the immobile phase. So this is PFAS that has sorbed to the sediment, but doesn't want to leave. It really sticks to the sediment. And we see two different plots on the right. One is for the carboxylic acids, one is for the sulfonates. On the x-axis is PFAS of different chain lengths, so shorter chains on the left and longer chains on the right. And we see that there's a breakpoint, a general breakpoint in how these compounds distribute. For carboxylic acids, we see that for chain lengths of 8 carbons to 10 carbons, so from PFOA and above, the red line, we see that it's really those compounds are the ones that have the ability to sorb and remain sorbed to sediments. For sulfonates, we see the same pattern, but the pattern is shifted slightly to the shorter end. So what we see is for PFHXS, so 6 carbons and longer, they have the ability to sorb and remain on the sediments. And so this is telling us and demonstrating directly that the shorter chains generally don't sorb in the sediments. They want to stay in the aqueous phase, and they're of less concern to the sediment sites. And then we also need to recognize that precursors and other related compounds, because of their larger molecular weights, because of their greater complexity, they tend to be more sorptive. So they're of greater concern for sediment sites. So a little, how does this actually look in the real world? If we look at some real world data, how does our understanding match with what we see? The EPA has done a fair amount of work in the Coosa River Basin, which is down in Alabama and Georgia. And the Coosa River has some diverse sources of the PFAS to the river, primarily as textile mills, but they also have wastewater treatment plants, commercial, industrial, and residential runoff. So if we look at some of the data they collected, this happens to be data for the Chattooga River, which is a tributary of the Coosa. And if we look at one location where they collected some surface water, it's shown in this pie chart. We see that the distribution of PFAS present in surface water is dominated by short chain carboxylic acids. That's the blue blob in this pie chart. If we compare the same location for the sediments, what we see is the sediments have a much larger fraction of long chain sulfonates, the yellow, and long chain carboxylic acids, the orange. This happens to be just for one location in the watershed. If we look at it across the watershed, across all the samples that EPA selected in this river, what we see is that that one sample location happens to be pretty darn characteristic. In the surface water samples, we see that it's dominated by short chain carboxylic acids, exactly what we'd expect based on our understanding of structure and partitioning. In the sediments across the watershed, we see similar patterns where the primary PFAS present in the sediments are the longer chain sulfonates and carboxylic acids. We do see some short chains in the sediments, but what we would expect to see over time as sources diminish, as the use of PFAS diminishes in the watershed, is that those short chains would be relatively quickly flushed out of the sediments because of their ability to partition into the aquatic phase. So to summarize and hit some kind of key points, some take-home messages for today for PFAS and sediments. Number one is we really need to understand our sources, where they're coming from, what's going on. And we need to have a detailed understanding of the structure of those sources, because we know that fate and transport is linked to structure. When you're building your CSM, when you're building your mass balances, trying to understand what's going where, we need to make sure that we understand transformation processes can be a key factor controlling PFAS concentrations in the environment. Not all PFAS are going to sorb strongly to the sediments. Really what we're focused on is the longer chains and the more complex molecules. And lastly, we also know that there's not a lot of sediment regulatory thresholds out there yet. That picture is really, really unfolding. It's kind of an ongoing area. And with that, I will hand off to Rob and let him discuss some more of the regulatory picture.
As when Ronald Reagan defeated Jimmy Carter, there was a lame duck session of Congress, and they felt like we had to pass a piece of legislation before Reagan came in and was likely to veto this. And that's when Superfund got passed. By anybody's assessment, it was an ill-fitted, drafted piece of legislation to address a known problem. And Congress tried to correct that in 1986 by the Superfund Amendment and Reauthorization Act. And that just, in my mind, created even more confusion. And I'm going to give you one scientific aspect of this, which is a great analogy to what's going on now with PFAS, and that is Congress mandated that the National Priorities List have 400 sites on it. So in 1982, Congress developed with EPA and published a rule, the hazard ranking system. And to get to the 400th site to be listed, the score was 28.5. Nothing magic about 28.5 other than it met the congressional mandate of listing 400 sites. Well, since that time, that has been the standard for whether a site gets listed on the MPL. To me, that's not scientific. So I was an adjunct professor at law school for 25 years, and I used to tell my students all the time that the most important laws are not those that are enacted by Congress. They are not the regulations that are published by and promulgated by regulatory agencies. They are not those that are created by the courts. The most important law is the law of unintended consequences. That's what happened with Superfund, and I see that happening again with the way we are dealing with PFAS. So let me start by saying that I don't think that there's much dispute that there needs to be some regulation of PFAS. It is a compound, a series of compounds that are ubiquitous in our environment and have potential health effects. So nobody is saying that it doesn't need to be regulated. The questions are how, when, and where. And the danger that I see is that we are in a headlong rush to regulate this in a completely uncoordinated way, which is going to exacerbate what I just said, which is the law of unintended consequences. So let's start with the science. And I'm going to build a little bit on what Miranda said, which is at this point, we are making regulatory determinations in a field where we have, in my mind, undeveloped epidemiological evidence. If you just look at the list of diseases that federal and state agencies have associated with exposure to PFAS, it is extremely broad. If you were going to bring an individual case, you would be challenging some of that epidemiology on Dahlberg standards. It includes low birth weight, accelerated puberty, testicular and kidney cancer, liver effects, immunity effects, and believe it or not, cholesterol changes. So if we're going to make decisions on how, when, and where to regulate this, we have to have proper scientific underpinnings of the human health effects. So that's number one. If you look at, and I'll get to this as a regulatory development in a little bit, if you look at the listing document, the proposed listing document that EPA has come out with, and also look at for hazardous substances under Superfund, and if you also look at under the Clean Water Act, Safe Drinking Water Act, the proposed MCL for PFAS. This is what the agency says, is that studies indicate that exposure to those contaminants above certain levels may result in adverse health effects. In my mind, that is not a proper way to regulate and set standards. I think Miranda alluded to this. We have even less information in terms of the ecological risks associated with exposure in sediments for exposure to the various PFAS compounds. So what we have then, and that uncertain scientific background or underpinning, is that we have cleanup standards being proposed or drinking water standards that basically say there's almost no safe level at which you can have exposure to these PFAS compounds. And if that is true, given the data that we have seen, at which most Americans, the vast majority of Americans, either in private or public water supplies, are exposed to PFAS concentration limits above those proposed standards, it would mean that most of the country is exposed to unsafe levels in their drinking water. So what's the consequence of that? The consequence of that is pretty profound. Now, I'm going to just talk about a very practical one. So let's assume that that's the standard and that there has to be remediation based upon that. We do not have enough activated carbon in this country to treat all of the public and private water supplies that would be above the proposed MCL level. That's just a very practical suggestion for that. If you're going to try to incinerate waste with PFAS in it, we don't have enough incineration capacity in this country to do that. Now let's look at the economic impacts of that. On the public side, who are the people who potentially could be spending money to deal with these issues? Well, we have public wastewater treatment plants. We have municipal solid waste landfills that are publicly or municipally owned. We have water utilities that could be either public or in some cases private. We have municipal airports. If those entities have to comply with cleanup of PFAS at these levels, that is going to be passed on to taxpayers, or in the case of private companies, potentially consumers. That is not an insignificant economic impact on this country. And I'll just give you one more as an example, and that is agriculture. If we have PFAS that is being treated in discharges to public owned treatment works, and you have PFAS constituents in the biosolids, those biosolids have been disposed of for agricultural purposes for years. So those sites may need to be remediated. And the agricultural industry in this country has potential liability for that as well. And then finally, many of these sites, as you know, PFAS constituents were used in firefighting foam. And so there are many, not just private airports or public airports that are owned by municipalities, but many of them operated Department of Defense operations on them. And so we also have potential liability for the United States government. So I'm not Chicken Little, I'm not overemphasizing this, but given the ubiquity of this potential contaminant in our economy and our drinking water supplies, if we are not careful in the way we coordinate our regulation, it can have significant impacts. And again, the law of unintended consequences. So this is just my 2 cents on this. There are sites that have very high levels of these PFAS constituents in drinking water. public drinking water supplies or private drinking water supplies. There should be a mechanism, either legislatively or by guidance or by enforcement authority, that deals with prioritizing those sites and not having this broad-based look at everywhere where PFAS could be. So that's kind of my overarching context for this. Let me just talk to you a little bit really quickly about what's going on with the regulatory agenda, just so you have some dates and some information. The PFAS MCL rule, which I just mentioned, which proposes a Safe Drinking Water Act standard MCL at 40 parts per trillion, is scheduled for promulgation in January of 2024. The PFAS TSCA rule, which will apply to manufacturers and importers of PFAS in products, has been finalized by the agency. It's not yet published. It's supposed to be published by the end of this year with an 18-month implementation schedule. So 2025 for when people have to start reporting under TSCA. Big one, designation of PFOA and PFAS, hazardous substances under Superfund, which would make this subject to Superfund liability, talking about potential significant liability on a broad range of parties without any federal intervention in terms of a legislative exemption for certain receivers of this material. And in addition, at sites that were closed for years that are in their second, third, or fourth 5-year review under Superfund could be reopened because PFAS was never looked at. That's, chilling to many people who've been involved at those sites. PFOA, PFAS, PRBS, and Gen X are proposed for listing as RCRA hazardous constituents. That could happen at any time. That is the precursor to listing them as a hazardous waste. And as I said before, we do not have enough disposal capacity in this country to deal with those substances as a hazardous waste at a RCRA hazardous waste facility. And then finally, there is a proposal that's scheduled for November of 2024 to add PFAS under Section 313 of the Emergency Planning and Community Right to Know Act, which requires annual reporting of the releases of PFAS to the environment. And that's scheduled again in November of 2024. I just want to, in the last couple seconds here, talk a little bit about PFAS litigation, because in addition to this regulatory structure that I just mentioned, which is coming down the tracks, there's a lot of different PFAS litigation out there. It's not controlled by a regulatory agenda. It's controlled by the people who are the plaintiffs in the litigation. The targets that we've seen so far: manufacturers, site owners of sites that are contaminated, people who generate waste that are sent to those sites that are contaminated. The plaintiffs are a wide variety of plaintiffs. States have brought these actions. Water utilities who think that their water supplies have been impacted have brought these actions. Individuals have brought them as personal injury plaintiffs or property damage plaintiffs. Those individual actions have either been brought in individual capacity and they've been brought as class actions. The causes of actions that are asserted are very broad. Some of them are brought as natural resource damage claims. Some of them are brought as traditional common law claims. The common law claims are tort claims. They range from negligence to strict liability claims to, believe it or not, consumer protection claims and failure to warn claims. So on top of the regulatory agenda that I spent some time going over and the dangers of that, that is, obviously partly a political process. This is a completely separate process that has its own liability that is not controlled by the regulatory process at all. So I guess my takeaway is we are at an inflection point. We have a known potential problem to deal with. This is the time to take our time to get it right, to coordinate it, to minimize the law of unintended consequences as much as we can, and rushing in headlong without that coordination, I think, could have profound negative effects.
All right. Thank you, Rob. That was really inspiring. And we do have about 10 minutes left to take questions. Before we do turn to questions, though, I want to apologize that the recording device only turned on after Rob has started talking. So we are going, Jared and I are going to re-record our sections and distribute to all registered participants, both the recordings. So Rob's as well as mine and Jared's together. In addition, if you tried at the start of the call to connect to the links that are in the resources, they were disabled. They've since been enabled, so you should be able to access them now. During the call, there were a couple of logistical questions that called those things to our attention. In addition, there was a question posed that asked why the evolution of PFAS production moved from the longer chain molecules to the shorter chain molecules. That change in production has primarily been driven by concerns about ecological and human health risks, where the shorter chain PFAS are generally believed to be far less harmful than the longer chain. This is illustrated through the difference in the MCL, the proposed MCLs. For instance, for PFOA is 4 nanograms per liter, while for PFBS, it's 2,000 nanograms per liter. Next, I'm going to let Paul Pershing talk. He's had his hand raised for a bit. Don't forget to unmute yourself, Paul. You're on. Paul?
Can you hear me?
Now we can, yes.
Okay. I put a comment in the Q&A, and I really is just wondering if there's any more details on how EPA is going to handle Superfund liability for PFAS. I know there was some talk earlier in the year that there was supposed to be some kind of an enforcement policy coming out this summer, and I don't think that's ever been released unless I missed it. But do you have any more details on what direction they're going, or how they're going to handle this situation?
Yeah, so there was actually a statement made by a senior official in the enforcement branch of EPA that indicated that they were going to try to use their enforcement discretion not to take enforcement action against public, what I would call utilities. So public airports, not private ones, public airports, municipally operated, public landfills, public wastewater treatment plants, and public water utilities. That is just an enforcement discretion. Obviously, it is not legally binding on them in any particular case. And more importantly, if the agency does not take enforcement discretion, that does not stop a private party who is a PRP at that site from taking an action against that party. So it provides, in my mind, a very small benefit to those. It is not insulating. It is not a defense. There are discussions that are ongoing. I think it's been, widely reported that certain of these classes of parties who deem themselves to be receivers of this and innocent in this process are trying to see if there's a legislative fix. There are a number, as you know, of exemptions to Superfund, some that were in the original, or defenses that were in the original act, and then some that have been engrafted in 1986 and the late 90s, and in 2002, I was involved in the Brownfields one. And so that may help some of what I'm talking about. And that's exactly the kind of thinking that is needed. Who are we really trying to target here? We can't target everybody. Who really should not be a target of this? But that is not, we don't have that yet. And we don't even have something other than that statement that they would exercise their enforcement discretion.
Thanks, Rob. There's also a question in the Q&A asking Jared whether a passive sampler, like a POIC, should be used to collect stream samples of surface water over the course of several weeks.
I think that's it's a great question. It really depends on what you're trying to ask with that sampling. I mean, it may be a blessing or a curse, but the PFAS analytical methods are actually really quite good. You can usually get down to sub-nanogram per liter concentrations. So it's not a matter of, detection limits. And I think that if the point in time, point in space grab sampling approach isn't answering the questions that you have, that, a passive sampler may be a good approach.
Great, thanks, Jared. Matt McCollum has asked in the Q&A whether there are any regulations for how to handle dredged sediment with PFAS contamination, any known timeline for when EPA will come out with such regulations. No, although I suspect that the Department of Defense, if they have not already developed sediment benchmarks, they are probably going to be the first to do so. So the federal government and state agencies are pretty far ways away from developing dredging benchmarks for PFAS, and indeed for many other sediment contaminants.
Can I weigh in on that question?
Oh, definitely.
This kind of highlights my point. So, there's contaminated sediment sites around the country that are being remediated right now under Superfund, not even a remedy selection. So that material is going to be dredged, hasn't been sampled for PFAS at this point, going to go to different disposal facilities. And who knows whether those disposal facilities are going to have liability for whatever comes out of that contaminated sediment, and whether people who had responsibility for the PFAS in the contaminated sediment will now have derivative liability at those other sites. Another example of the law of unintended consequences.
Yeah. And Paul asked whether he can get a copy of the presentation slides. Yes, we will distribute them with the links to the recording. I'm going to allow Yamini to speak. She's been patiently waiting with her hand up. And don't forget to unmute yourself.
Hi, Miranda. Great presentation. Thank you so much. I actually did not realize that I raised my hand, so I have no questions. Sorry about that.
No worries. Matt, you've got your hand up, and I will allow you to speak. Matt, you can go ahead. That may be a residual from your question. All right. I think that we're just about at time, and the stream of questions has slowed down. Certainly, if anybody has questions for any of us after the webinar is over, you should have our contact information down at the bottom of the resources. Don't forget to go there and download them. Feel free to email any of us. Watch also for an email containing the slides, as well as links to the 2 recordings. It'll be a few days since Jared and I need to re-record, and we appreciate your attendance. Thanks a lot.